Hi, my name is Beau Jones. I'm a paleontology student here in Edinburgh, but I want to tell you a little bit about a research project I did probably like 2 summers ago now. So it's been a while since I've looked at any of this material, so apologies if I don't really know what I'm talking about. But it was a project that I took on, which was funded by the Paleontological Association in conjunction with the British Antarctic Survey. So they sent a load of people out to collect fossils, and then shipped all these fossils back and handed them off to me to see what I could do with them. And I've promised you here a little bit of paleoecology, and I've also promised you some biomarker stuff, which I have redacted because even I don't really understand it, and it's been 2 years, and I didn't want to subject you to lots of big graphs where I point at spikes and go, yeah, that's pyroline, it's a compound, and it just wasn't necessary. So this is much more fun and creative, and you can actually look at some funky fossils. So it's a bit of an overview. I'm going to tell you about the KPG, the geology, paleoecology, a bit of the prior research, let you know what's been found, and then what we did. Well, not me. I wish I'd gone to Antarctica, but I didn't. And then some of the preliminary results that we've got. I obviously have left Bristol now, and I'm no longer working on this, so they will remain preliminary for the foreseeable future. So yes, this is the geologic context. We're looking at the KPG, which is the Cretaceous-Palaeogene boundary, as was said earlier. So this is the period where famously all the dinosaurs died. So we have this massive faunal overturn where everything changes. You go from these massive dinosaurs, these huge things, all like cold-blooded or mesothermic, depending on who you're asking, to mammals and your birds, and suddenly you have this massive radiation of flowering plants that are just going absolutely bonkers and changing everything that we know about our environment. So we really want to investigate this environmental change and see what was going on and why. So this is a bit of the paleogeography. The big meteor, which I'm sure you're all very familiar with, it's famous in Fantasia. Oh no, it's not in Fantasia, but we all know about the big meteor that hit the dinosaurs and they all got wiped out. So this hit the Gulf of Mexico, and then all the way down here in the Antarctic, that little blue circle there, that's Seymour Island, which is the island that we were looking at to pick up all of these fossils. And you can kind of like watch the KPG all around the world. It's one of these amazing sort of like events which there is markers which are just completely global. So you get this iridium layer which shows up pretty much globally. It's harder to find in the Antarctic because it's further away from where this site was. And the same goes for these impact spherules. So imagine this big meteor hits the Earth, sends up a load of impact ejecta, and as it's flying through the air at like insane speeds, it heats up really, really hot, and all of that quartz that's been blown into the atmosphere melts, solidifies into these globules, and then falls back down to the Earth. So you just get these little sort of almost like frog spawn rocks, where if you look at them underneath a thin section, they have these amazing glassy globes, and sometimes they even contain atmospheric water. So they kind of act as little what they called snow globes. So you have these amazing sort of like captures of life 66 million years ago. So this is Seymour Island. As you can see, it's just on the little sort of like tail end of Antarctica there, and right above it is South America. And this is the island that my supervisors were working on. You can see over the side there, just here, it has a massive cross section that takes you all the way from the end of the Cretaceous up until the Quaternary, which is us now. So it's really, really great for looking at stuff within sort of this period of time. And you can even see just down that little line there, there's a nice little dotted line of where the KPG is. That's the point at which the Cretaceous and the Paleogene are meeting on the island, and you can go there to study what's going on. So quite a lot of research has actually been done on some of the really cool things that have been found on Seymour Island. So lots of big titles that say the oldest or the biggest, or you know, the most toothy or the apex predator. You know, there was a lot going on on Seymour Island during this time period. It wasn't this like barren place that had nothing going on. It actually has an incredibly diverse vertebrate assemblage, which just everything was living. It was alive. But you know, this is the vertebrate stuff, and perhaps slightly less charismatically, I'm interested in the invertebrate stuff. And the person who did this research is called Zinsmeister. And the way that he would do this and go about looking at these marine things was he would go for a little bit of a dawdle around the island. And as he was walking, he would spot fossils. And for each fossil that he found, he would just drop a little GPS location on his device, giving us this lovely set of coordinates. And although this is really great and it gave us an amazing view into the kind of things that were living in Seymour, or rather around Seymour Island, during the Cretaceous and the Paleogene, it's not necessarily good science to just go on a dawdle and spot things and pop them in a GPS. So it kind of gives you an idea of like what is here. And you can say there's 40 bivalves and there's 3 ammonites, but it doesn't actually give you an idea about how they're coexisting within this community. So that's really what we wanted to do. And a lot of the research that has then been used with this sort of like throwing away GPS coordinates and looking at sort of like where things have spottedly been around has given rise to this idea that there's a 2-phased extinction on Seymour Island. So each of these little numbers is a different species, and each of the little dots is an occurrence. So we've said, we've got one here, we've got one here, we've got one here. And you can kind of like start to build up an idea of how long they've been around. And so Tobin, which is this fella, suggested that there was a second extinction happening just before the KPG, which was caused by Deccan trap volcanism, which was a thing. It did happen, and it was pretty extensive and caused some pretty serious global climate change closer to the site, which I think was in India or thereabouts. Might be wrong. Haven't done it in 2 years. Got to keep that. Got to remind yourself I haven't done this in 2 years. And we looked at this and we thought, right, well, fair conclusion when you look at the data. But there is actually something which might explain what this person is finding, which is called the Seymour-Lipps effect, which is, if you can see this sort of like big section here, if you imagine that this is like the entire rock record, the whole of a section in Antarctica, and then this tiny little slither here is the ones that you can go and actually capture in that 2 or 3 weeks that you're on the island. Even though you get all 3 of these species existing within the rock record, because you're not sampling them, it looks as though they have a staggered extinction. So it looks like things are going extinct a lot earlier than they actually are. And we think that that's what is causing this effect. They might just be rarer, or we might just be looking in the wrong place. So we wanted to go and see what we could do to clear up this kerfuffle. So this is the team. These are my supervisors, specifically this cheeky chappy here, which is Dr. James Witts, now at the NHF, which is very exciting. And so they went out onto Seymour Island and they sampled these 2 locations. First one being Lahilia Hill. I will tell you about Lahilia later. They are the bane of my existence. And then the second one is Filo Negro, which means the black rock. And these are 2 of the sites which a lot of this work has been done on in past, where people have been going through and just finding things. So they went out and they decided to do systematic sampling, quantitative sampling. So they went to these individual sites and they would work up very systematically and take in samples, bag them, and then ship them all back to Cambridge for me to pick up and mess about with. So yes, these are some of the cool things that we found. This is always the bit that I like the most. These just, the exciting thing about doing any research is looking at beautiful things for as long as you can. And so, like I said, Lahilia, This is a massive bivalve, which is very, very common on Seymour Island. You've got these amazing ammonites like Maurites, and earlier that sort of paperclip shaped ammonite, which I showed earlier. This is Diplomacerus. And then there's a number of slightly less common things. So we do get marine vertebrate, marine reptile vertebra, which are fairly sizable and amazing to hold. And a number of interesting gastropods. I think this one, Struthiogenopus, is so interesting because it's got this sort of like big flange, which it snapped off in that picture, but you can get them with these big, amazing flanges. If anybody wants to ask me later about mesofossils, which are these kind of like intermediate size fossils, I would love to talk to you about them. But the thing I didn't realize when I went into this project is that even though this looks really sort of like flashy and cool, is that I would be spending quite a lot of time working and working and working and working on these annoying little things called Rotularia. So by the end of this project, it was only about 2 months that it took to do the whole thing, sort of speed running it. But at least 3 or 4 days of my life was spent staring at just bags and bags of these little tube worms. And annoyingly, in the final data, we cut all of them because we thought it wouldn't be a good idea to keep them all in because it would skew the data. So I've wasted 4 days of my life. So this is what I did. I got all of these bags of crap from Cambridge, and I sat down and I just stared at them and I identified things and I put labels on them, bagged them up and sent them back off to Cambridge. But as I was doing so, I was sort of like trying to work out what each of these animals would have been like in life. So I sort of very systematically went through and I quantified what all of these things would have done. So this is what we call a mode of life. It takes into account how you move, how you eat, and what eats you. And in the end, we have this massive, ridiculous spreadsheet. This doesn't even begin to look at all of the things that we put into this, but I'm very proud of it. And in the end, we came up with this. So we took all of this data, and then for each of the individual sections, as you can see on here, so these are the different sort of like layers running through, we quantified them out of 100 to just kind of see what was dominating and what was being a little bit more passive. And immediately you can see that there is a crazy difference between the Cretaceous and the Paleogene. So there's just clearly been this something which has just caused the entire community to collapse into anarchy. And I just want to take you through kind of a couple of things that we had in these preliminary results. So Lohelia, I did tell you that they would come back. Lohelia are what we call a disaster taxa. So when you go out onto Seymour Island, if you ever get the chance, when you're walking, you will be walking on a pavement of these things. They are so abundant after the Cretaceous-Palaeogene mass extinction that they just started sort of like chucking them into piles and sort of being like, oh, just tally them, next one, and just tally them and move on. So we know that there's a lot of these. And so clearly there's been something that has allowed this group of bivalves to just explode and go absolutely mad in their radiation. So that is a pretty good indicator that this was like the big thing. There was a single massive thing that just blew everything out of the water. And these guys kind of are our indicator for that. And then this is another really interesting bar that I thought I was pretty surprised when it came out, but then I remembered all of the time that I'd spent counting these annoying little things. And then I was like, actually, no, that makes sense. So these are Lammatula. They're about this big, and they were sort of like condensed into this one layer and would sort of stack inside of themselves. And it was my job to sort of just like push through all of them and just count all of them up, but then they would break. And then I'd have to sort of try and work out how to quantify them and be like, oh, well, you know, there's two sides to a shell. So if I have one shell, is that one guy? Or if I have two shells, is that one guy? And it just all got a little bit complicated and we're not quite sure what we're going to do about that yet, actually. But yeah, I thought this was really interesting that these guys are sort of like, even though they're so tiny and you could probably fit about 600 of them inside one lohelia, they're playing such a big part in our data. So it kind of gives you an idea that even though there's a massive amount of something, it might not be playing as big a part in the ecosystem as you think. And finally, this we did end up removing from the final data, but I still thought it was interesting because there is a trend there, is these sort of like casts or fragments or sort of unidentified gastropods. I'm not quite sure what I think about this, but I remember thinking it's interesting that they go so bonkers and then immediately get cut down, because either there's something to do with how they're being preserved. So either this sort of like shallow marine sea has gotten really turbulent and it's been breaking them up, or they just poof out of existence at the KPG. So I thought this was interesting, even though we did end up taking it out of the final data. So this is what we published, just a little something to get you interested before we get into the nitty-gritty of the biomarkers, which I won't force you to suffer through. But we just we combined those two KPG sections because, as you can see, they're actually quite similar. And then put all of these lovely trophic group generalizations on so that people could get an idea of what we're looking at. So yes, these are my take-home points from this project, is that this is a really high-resolution sampling from the KPG of Antarctica. It's a single massive extinction that we are seeing. We did not see any proof of a secondary extinction happening beforehand, but we did see this massive faunal like overturn. There was clearly a bit of sort of, as you can see, like things are moving, things are coming in, things are coming out, but there is turbulence, but it's not an extinction. And then finally, the biomarker research is ongoing and complicated, but we will get there eventually. So thank you so much for listening. These are my amazing supervisors who I miss every day. And if anyone has any questions, I'm happy to answer them.